The antibody-mediated immune system revolves around B cells. That's because B cells are the only cells that can produce antibodies. Antibodies are fairly large proteins that are produced and attached to very specific antigens. There's millions of different B cells that are produced by your bone marrow, just like there's millions of different T cells. And each of these cells varies at their receptor, their antigen receptor. So just like T cells have different T cell receptors, B cells have different B cell receptors. And these receptors are specific to any amino acid sequence that's different than those amino acid sequences in our own proteins. So this is how our immune system recognizes foreign things by having receptors for foreign things called antigen receptors. So the activation of a B cell occurs when a microbe enters the body. This microbe has to be an extracellular microbe, so it's found in a body fluid. It could be found in interstitial fluid, lymph. It could be found in the bloodstream. It could be found in other secretions, like digestive secretions. So when there's a microbe present in a body fluid and a B cell recognizes it using its B cell receptor, so here's the microbe, here's the B cell, it's got a receptor that can identify that little part of the microbe. That little part is often called an epitope. An epitope is the exact, usually amino acid sequence that an antibody can bind to, can stick to, and recognize. Once that happens, there must be co-stimulation. So notice again, there's this requirement of a helper T cell to co-stimulate this process. So helper T cells co-stimulate cytotoxic T cell activation. They co-stimulate themselves, and they co-stimulate activation of B cells. So that's shown right here. Here's the helper T cell co-stimulating using several interleukins such as interleukin 2. This leads to proliferation or cloning of that specific B cell. So we're not going to clone all of our B cells. We're going to clone just the ones that have B cell receptors that can recognize specific epitopes on the pathogen that's present. This proliferation leads to cells that are going to be secreting large amounts of antibodies. Those are known as plasma cells or plasma B cells. So plasma B cells are the B cells that were activated, cloned, and are releasing the antibodies. They're secreting antibodies. These antibodies go into body fluids. So if this B cell was in a lymph node, and this microbe got brought into the lymph node through our lymph, that B cell is going to stay in the lymph node and just secrete out antibodies. And those antibodies are going to enter the lymph. That lymph eventually goes into your bloodstream at the subclavian veins, and now you got antibodies in your bloodstream. And once you have antibodies in your bloodstream, they can go anywhere. They can go almost anywhere in the body, and they're all going to be specific for that microbe, and they're going to attach to that microbe. At the same time, we're going to make memory B cells, just like we made memory cytotoxic T cells, just like we made memory helper T cells. We're going to produce memory B cells. And those cells are going to be there, so if this microbe enters another time, maybe years down the line, we've got more cells present that have receptors and antibodies against that microbe. This graph just shows us the difference between the first time we're exposed to a new pathogen and the second time we're exposed to a new pathogen. So it's called primary response versus secondary response. So notice here at time 0, we got exposed to a pathogen, or we got vaccinated, because a vaccination is mimicking a first exposure to a pathogen. And in response to that exposure, we're going to make a certain amount of antibodies. Usually the first antibodies produced are from the class called the IgM antibodies. IgM antibodies are large antibodies. We'll take a look at a picture of them in a minute. And they're usually the ones first made. Then there's what's called a class switch. The B cells switch over to what's called IgG antibodies. And those IgG antibodies are produced. So hopefully during this time period, we get better. So if this was the flu virus, hopefully we're okay 14 days later. Or maybe this is a vaccination and you don't get sick from vaccinations, but you still have this immune response occurring. Then a couple more weeks go by and you get a secondary exposure. So maybe you had the flu and now you got exposed to that same flu virus again, or you had the vaccine and you get exposed to the flu virus. That second exposure, you're going to have that IgM, but notice the IgG. 1,000 times more IgG antibodies will be secreted by B cells. And that's because you have those memory B cells that have already been activated. They already know what that pathogen looks like, and they'll produce large numbers of antibodies. The amount of antibody in blood is often called a titer. So antibody titer is just a measure of antibodies in someone's bloodstream. It's very useful to just see what's going on with the immune system. If someone's immune system is suppressed, they won't be producing large numbers of antibodies when they're exposed. If someone has an overactive immune system, maybe an autoimmune disease, we can measure antibody titer of what we call autoantibodies. Those are antibodies that are attacking some component of yourself as opposed to a foreign entity. This table shows us the different classes of immunoglobulins, aka antibodies. So remember, immunoglobulin is just another name for an antibody. There's 5 major classes of antibodies. And there's a lot of information on this table, and I'm going to try to focus you in what's most important. But first of all, there's a letter designation. So Ig stands for immunoglobulin. And then the big letter is the different classes: IgG, A, M, D, and E. Notice they all have this kind of Y structure. This is a protein, so it's a large protein. So these are amino acid sequences. And at this part of the antibody is where the antigen binding component is, here and here. So this single antibody, this kind of Y shape, can recognize the antigen here and here. This guy, IgA, is a dimer, so it can recognize in 4 places. IgM is a large pentamer. It's got 5 similar structures. And then D and E are both monomers. So they look a little different, at least these two, A and M. They function and their characteristics are slightly different. IgG is the most abundant. So when you're sick, you've got a bunch of IgG antibodies circulating in your blood primarily, looking for that microbe. So these are the ones that really are prevalent when you're fighting an infection. How do they do it? That's a good question. Well, one is they enhance phagocytosis. We learned that before when we looked at complement. We call that opsonization. So antibodies also enhance phagocytosis. They can attach to toxins and neutralize toxins. There's a number of toxins that get produced by bacteria, and it's the toxin that makes you so sick. So these antibodies can actually attach to the toxins and reduce that toxicity. They can also help trigger the complement system. So the idea is an antibody sticks, right? It attaches itself to antigen, and that can help tag that pathogen. It can help neutralize. It can help immobilize. So we can make antibodies to a bacterium's flagella, and it can't move around in the body. We can make an antibody to a virus and prevent that virus from attaching to a cell, because viruses, in order to infect, have to attach to the cell first and then enter. So antibodies are very useful in blocking pathogens. IgAs are found in certain types of body secretions: sweat, tears, saliva, mucus, breast milk, and GI secretions. So they're more present in certain secretions. There are a little bit in your blood and lymph, but they are also found in these other fluids. IgMs, two things I want you to remember about IgMs. One is they're the first antibodies produced, first antibody class to be secreted by plasma cells after initial exposure to an antigen. So they're always kind of the first one. So you can, if you see a lot of IgMs, that means that it's a pretty early immune response. The other thing I want you to remember is, remember back when we were doing blood typing, and we were talking about, the problem is when the recipient can make antibodies against the donor's antigens. Well, those antibodies are actually IgM antibodies. So the anti-A and the anti-B antibodies that determine blood compatibility are these larger pentamers. IgDs are surface receptors on B cells. So if you look back at a B cell and saw those B cell receptors, those are actually called IgD antibodies. And then finally, IgE antibodies are involved in allergic or hypersensitivity reactions. So these guys are located on mast cells and basophils. And when they bind to antigens called allergens, it activates an allergic reaction, which we often call hypersensitivity. So people with allergies have a lot of these IgE antibodies kind of at the root of that inflammation and that reaction. They also have been found to help protect against certain parasitic worms. So those are the 5 classes that makes a good note card. Make 5 note cards, the name on one side, what distinguishes the 5 on the other side. Go over them a few thousand times and you'll be good to go for the different classes of antibodies. The next video, we're going to finish up the PowerPoint, talk about some of these other things that are involved in development of your immune system. This figure looks scary at first, but we're going to kind of go through it slowly so we understand a couple of the major processes that happen in your bone marrow and your thymus in order to develop a nice, powerful immune system.